Hello, Assalamu alaikum. In this video, we will understand about the cellular anhydrobiosis. The cellular anhydrobiosis is the loss of water is known as desiccation or dehydration. So it can be reversible when we will hydrate with the water. So remember the water is important of life. All metabolic reaction is possible due to the water because the water is the excellent solvent for the solute. The transportation of content is the possible due to the water. So the hydration is too much important for the normal function. But in this video we will understand about the dehydration and how it can be benefit or harmful. So this is the bacteria. The high temperature and high solute in the medium will lead to dehydration of water, and in this the prokaryotic cell will activate that gene which that resist the dehydration condition and survive. While the eukaryotic, like human, which the loss of water and exchange of water and balance of the water is very important. But the high temperature and the fasting with the restriction of water will lead to dehydration and due to the respiration and the perspiration as well as the sweating, and the cytosolic water will be deficiency inside of the cell due to the osmosis. The semi-permeable membrane allow to move water only, and diffusion is no permeability. Water moves toward high ionic salt. If the hypertonic condition are present in the blood, so in this way the water will move toward the blood, and the shrinkage of the cell will possible in the RBC or other cell. The transportation can be possible moves water and sodium by the using channels like aquaporin 2 channel, and the ATP is also used. The environmental condition is the hypertonic, isotonic, and hypotonic. For example, the hypertonic means the hypotonic condition inside of the cell. The water will move outside because the hypotonic to hypertonic condition because the solute when increase in the environment. For example, the isotonic mean no any changes. So the inside the cell and outside the cell is the environment same. So ATP is required for the sodium influx as well as the water balance. Hypotonic condition mean the hypertonic condition inside of the cell while the hypotonic in the environment will lead to water move inside, and in this way the water uptake can be increased due to the hypertonic condition, the solute increase. Let's understand about hypotonic condition is due to the hydration. This is the osmotic stress in the cell. The hypertonic condition in the environment. Water will be less. In this way, the sodium, the solute, will be decreased inside of the cell. The hypotonic condition will be formed. So in this way, the water will move outside due to the hypertonic condition, and in this way, the hypertonic to isotonic formation and the sodium when increase, the water will be decreased. The stress or desiccation will occur. The sweating will lead to also cause dehydration due to the hot temperature, hot weather. The stress and desiccation will lead to in the hypertonic condition will lead to activate that genes which that is important for the metabolic resistance, inhibition and removal gene. The metabolic resistance gene activation will occur, and in this way the combating with the harmful environment. In this way, the apoptosis and total lack of metabolism will occur due to the anhydrobiosis. Metabolic dysfunction and shrinkage and injury of the cell will lead to cause apoptosis. So in this way, the cell proliferation, differentiation, and division will be impossible. So in this way, the quantitatively definition of the anhydrobiosis is the condition in which the water content is less than 0.1 gram, 0.1 gram, the H2O per gram, the water per gram, dry mass. So in this way, the cell division will decrease and differentiation will decrease, will lead to basically will lead to inhibit the cancer causing cell, and in this way the short duration of the anhydrobiosis is very important during the fasting with the restriction of the water. So the less water will get the benefit more. The anaerobiotic state, the survival of the organism, is possible. How it is possible? The gene basically activate will lead to solute increase and enzymes increase during the hypertonic condition of the cell will lead to use the energy via a glucose metabolism. And in this way the glycogen formation will be increased. When the glycogen formation will increase will lead to less water requirement and little bit glucose will use to produce ATP. But remember, the when hydration stage, when we will intake water again, the water will move inside due to the more glucose level, and in this way the glycogenolysis by enzyme gene activation during the again hydration will lead to more uptake of water, and the solute will increase and double for the uptake of the water. This solute doubling is due to the gene activation during the stress, the biological stress. This is so important. Let's understand about the cellular metabolism in the anhydrobiotic state, the experimentation. So let's understand about how we can identify the genes activation and how it is possible. This is the prokaryote. We will learn from the prokaryote, which that contain DNA, and DNA contain gene. So this during the stress, the biological stress, the anhydrobiosis, the gene will be activated to produce messenger RNA. This messenger RNA character is synthesis of protein which that will resist during the anhydrobiosis and produce ATP and normal function. The reverse transcriptase in laboratory we will use after the extraction of the mRNA to convert the RNA into the DNA via a reverse transcriptase enzyme. After the detection, after the amplification in the PCR, the real-time PCR and the microarray and RNA sequence will lead to a check in the gel electrophoresis before stress and after stress. So we can identify the new gene activation for combating the challenged, and in this way the new gene activation and the gel electrophoresis, the environmental condition will lead to identify the new gene activation. For example, this mRNA can be a new mRNA which that is before the anhydrobiosis absent. So the enzymatic activity in the anhydrobiosis state is example phosphorylase. Phosphorylase is the, for example, enzyme produced for increase the energy generation in the anaerobiotic state, and the pH futile cycle turn our macromolecule, for example glucose, into the glycogen. Motility, ATP hydrolysis solute increase, decrease aquaporin channel gene activation and increase water homeostasis, but also it can be increased during the hydration time. So increase the salt absorbed in channel protein and heat shock protein during the anhydrobiosis, and the anhydrobiotic resistant protein due to the mRNA transcription will lead to cellular anhydrobiosis resistance can be possible. For example, the inhuman, when dehydration during the fasting of the water and restriction, a drought condition, will lead to new gene activation by cellular stress, will lead to adaptation by epigenetics. So the epigenome activation will lead to produce the mRNA. mRNA will synthesize the protein. This protein can be an enzyme, the functional protein, heat shock protein during the hot weather, and other protein which that will maintain the body, and the biological stress will lead to increase the longevity. This is the extracellular water will be when decreased due to the anhydrobiosis will lead to move water into the extracellular into the blood from the RBC, and the dehydration will occur, the intracellular water move out and stress will occur, will lead to recovery when we will hydration, when we will intake water, when we will drink. So the water when increased, again reverse reaction and the high order function will occur and normal physiological function. But remember that things, the gene activation will remain constant and work efficiently for normal homeostasis. And our body learn again dehydration combating system. Long-term dehydration decrease glucose level in blood and cell and glycogenesis formation, growth. And glucose decrease in blood will not bind with protein and function upregulation because the glycoprotein formation will be decreased will lead to normal function. In this way, the when water will decrease, will lead to again summarization, the genes activation, biological stress, and sirtuin genes activation will lead to longevity genes activation, and water fasting is the key to learn cellular mechanism to survive in dehydration state. While the glycogenesis, lipogenesis for less water solubility and glucose in blood will decrease, will lead to get more efficient work. But the longer period of the cellular anhydrobiosis will lead to apoptosis of the cell, and in this way the death can be possible. But the shorter duration of the anhydrobiosis or desiccation will lead to activate that genes which that is important for the cell function normally properly. And that is why the biological stress is important for the shorter duration, but the longer duration is not good. I hope you make sense about the cellular anhydrobiosis. In this video, we have discussed about the anhydrobiosis. If you like my video, please make sure to subscribe, like, and share. See you in the next video. Thanks for watching. Please make sure to subscribe and share.